Today we will discuss about this mechanosensing topic in terms of the pancreatic cancer. So we will cover how the ECM remodeling will promote the metastasis in the pancreatic cells. So this is just the same thing that we are discussing in some of our previous videos, that how the extracellular matrix or mechanosensing can uh, lead to the reprogramming of the cells. So in this case, the reprogramming occurs in a way that will promote the metastasis of the pancreatic cells across different parts of the body. So when the stiffness of this, uh, when the stiffness across the tumor cells increases, what it will leads to, the it will affect this arginine cycle process. So instead of arginine being converted into this arginosuccinate, then citrulline, thereafter orthinin, the arginines directly goes to this 1,2-acetate, and thereafter this creatine formations occurs through these CKB pathways, where the ATP productions is increased. That we can see that through this phosphocreatine, so the CKB pathway will lead to this phosphocreatine, and thereafter also again it will then converted into this creatine. So there this ADP will be converted into the ATP, and finally it will lead to this formation of this creatinine. So what the researchers that they have observed, that when the cells are grown, on a stiffer matrix, and also during the scratch assay, they can see that there is a high production of ATP, or the ATP to ADP ratios are high. And when they did the detailed analysis, or when they did the pathway analysis of individual metabolism sections that is related to the energy, they have find out that it's the YAP pathways, that is yes associated protein 1 pathways, that will lead to this high CKB expression. And this high CKB expression will lead to this phosphocreatine conversion again into this creatine, and there are followed by this creatinine. However, the pathways are more predominantly found that the formation of creatine is high, and this creatinine and this arginine cycle process is reduced. So from there, they also get information that ATP to ADP ratios are high. Thereafter, the ATP productions are high, and this could be due to the fact that which are at the cells which are in the stiffer matrix requires high energy to migrate from high energy for the migrations. And this is what they have observed in the uh, cells that are at the leading edge of the invasion. That can be seen here in this figure on the bottom side, that the cells that are at the leading edge will has this actin fiber formations, that is rod-like structures, and also there is a fusion of the mitochondria that has also been observed. And we have discussed these reasons why the fusion of the mitochondria proteins occurs, and this actin fiber rod-like structures formation in our previous video. You can refer to that video to understand these fusion formations of this mitochondria and this actin fiber rod-like structures formations. So this video or this research will come to a this research will in the end come to a conclusion that the the change in the stiffness of the extracellular matrix will also affect or will also affect or will also affect the cellular reprogramming within the cells in a way that it will promote metastasis. It will promote metastasis in the pancreatic cancer cells. And the other things that will also promote, that has been understood before this research, are this nutrient stress environment are also been exerted in the tumor microenvironment or extracellular matrix surrounding the pancreatic tumor cells, and that also leads to the cellular reprogramming in a way that will promote proliferations of these pancreatic cancer cells, and at the same time this metastasis, and also helps in the metastasis and migrations of the cells. So I hope you got a basic idea about how the mechanotransduction occurs in the pancreatic cancer cells in a way that will promote metastasis or migrations of the cells. If you need to understand more about this ECM remodeling and its relation with the pancreatic cancer cells, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.